Wait a minute.
I can't tell you right now. I have notes.
Was there a
I'm
question?
gonna just answer it first and then I'll.
You should have just said quiz day.
Mr. White, do we
Let's
submit it with the
Stop,
questions?
no.
You can't tell
What?
me.
The FRQ?
Yeah.
Yeah. Alright, so
She starts.
this is where we, about where we were, right?
Bro, how many people are in here? 5?
No.
3, 6, 7.
Oh, we went through Chargoff's rule, the practice FRQ, we should be on page 14 now. Right?
No.
Yes,
Oh yeah.
went through that, that.
Yeah,
Quick
14.
review, went through that. And practice FRQ. Oh, see another slideshow. Awful. So, DNA replication,
Like,
open sesame, it's not opening.
well, I mean, if there's enough students in your class.
So how does DNA go about replicating? Again, anything that ends in ASE in biology is a what? Enzyme, there we go. So we're gonna talk about a couple really important enzymes, DNA helicase, DNA polymerase. There's a couple others that we'll introduce when we start putting things together and repairing broken DNA. But DNA helicase starts everything. DNA, right, you've got your chromosome, and on this chromosome, it is this tightly wound double helix, right? And so this is our sugar phosphate backbone, and then our nucleotides in between, right? And then it's like wrapped up. So this is right. You have to open. You have to relax the DNA, and then you have to unwind the DNA. Then you take DNA helicase, and it goes through and it separates the two strands. Cool. That's what DNA helicase does. This is why that pickup line, if I were an enzyme, I want to be DNA helicase so I can unzip your genes.
Wait, can you repeat that so I can record it?
It's already recorded.
Okay, thank you.
Oh, no. It's that pickup line. You guys never heard that?
No.
Nobody's ever come up to you at a party?
No.
Yeah, let me unzip your jeans.
No. No.
No. No.
I'm
Never.
told you that.
Oh, nothing, dude. Fun words. We're gonna talk about leading strands and lagging strands. DNA is directional. Remember the 5' 3' ends, right? DNA is read in a specific direction. When it's read in a direction, then, right, one has to go one way, one has to go the other way. If you're going towards the replication port, you are on the leading strand. If you are moving away from the replication port, you are on the lagging strand. Lagging and leading can switch positions, right? You can be the lagging strand for this part, leading strand for this part. Just depends on what it is that's going on, where we're at. Alright, so DNA replication. Replicates during the S phase of the cell cycle, right? We have interphase. Interphase has G1, S, G2. In the S phase, synthesis, right? This is where we replicate the DNA. That should all be basic common knowledge and review by now, right? How does DNA replicate? We had to figure out what happens. There's a couple choices. DNA makes a copy of itself and sends that copy to the new cell. DNA, well, we can, if it's conservative, what we're saying is this gray is the original DNA strand in the cell, and it copies itself, and the original DNA comes back together, and the new goes over here. If that happened, then in cell division, if we marked the original DNA and then let the cell divide, then one cell, right, one line of cells would always have that original line of DNA, the others would be copies, and that's what we would see. Or it could be semi-conservative, where the DNA separates, here's a copy, and they each join with one of the original strands, or parent strands. Or dispersive, where little chunks are copied and it's like disassembled and then comes back together all jumbled up. Which one do you think we see? Conservative, semi-conservative, or dispersive?
Come on.
Or maybe
Dispersive.
We're gonna see semi-conservative. So that as it's split, right, when one's being made, it's sticking to this side, this one's sticking to this side, so that it totally unwinds, you still have two strands, one old, one new, one parental strand, one daughter strand. So models of DNA replication, conservative, the parental strands directly synthesis an entirely new double-stranded molecule, and we would get this, the parent strand is fully conserved or saved, unchanged.
Wait, where is that?
That's right here, you
Oh,
can
you
write it
write it?
here.
Oh.
Yeah, you can write it right under there. All you need for this is, right, the, parental strand, stays conserved or unchanged. The new double strand is what's synthesized, right?
Wait.
Oh my gosh, shorthanded.
Bruh. I told you, that's why I was like, wait, you should
Put your line in.
This is the only reason why.
What, you already heard it?
Yeah.
Are we good?
No.
I
What
would
do you mean,
not.
just take it off
I
of
will
this?
not leave my seat.
I already
even
never
if
get
it's
one.
for this
I genius.
class. I was telling my mom, I'm like, Mom, if you want to pull me out, you gotta pull me out anytime after second period. I have all.
Okay.
If I do my
I'm
thesis.
almost done.
Oh, no, 3.0.
Yeah, no,
think-
no.
I thought you said.
I thought you
I
said.
thought you said 2.6.
Oh.
I was
No.
like, that's no, you the classes right now, right?
No.
Okay.
Alright.
Semi-conservative model.
Agreed.
Two parental strands each make a copy of itself, and after one round of replication, the two daughter molecules each have one parental and one new strand. Yeah.
Actually, I would have been better freshman year.
Actually, yeah, cause like freshman year I had no hard classes. This would be
I
like
felt
the
like
only
in
hard
freshman year I probably would have cared more.
Yeah. Freshman year I was 4.0, bro.
Kinda hard to believe.
Freshman year I was so
No,
awkward.
it's not.
Scarlet. Scarlet, Mr. Kim.
Yo. He's the reason why I had a 4.0.
I've heard so many good things about this kid.
Nah, everyone in my class hated him.
That's okay, I'm not. He was good.
Is he a
One
math
good thing
teacher?
about this-
He got
Well-
fired.
Huh?
He got fired.
He did not get fired.
Or like he got laid off like all the other like COVID teachers.
Yeah,
I like this class.
I like the context.
yeah, there's a difference
I
between
like
fired
this
and laid
class,
off
it's
too.
just-
It's
It
Fired
makes-
means you
it makes
goofed
me
up.
feel really bad whenever I don't get this.
Right.
Makes sense, yeah.
Laid off is, hey, just gotta make cuts. That's what happened. That's what
Did
happened.
they find out who's getting cut?
Did we find out?
Yeah.
Well, yeah, there's still about like 60 teachers that are getting cut.
Like which teachers?
I don't know. I mean, we knew the 200, I don't know which of them had it rescinded. Rescinded means take back the, hey, we're gonna lay you off. Mine was rescinded.
Did you guys turn in the FRP?
I don't know if I'm staying or
Not
not.
yet. I'll turn it in.
Because I was on the coming to teach,
Did you guys do the,
Teach physics.
the case study? Yeah.
Well, yeah, physics. Friday, the school district in Oregon was supposed to go to, just laid off 18 teachers, so
Oh,
it's everywhere.
Oh.
Just because kids are not coming to school.
Oh, yeah.
there's a couple things. A, people are waiting longer to have kids. B, they're having less kids. And there's just, like, the attendance is horrible. You guys are 18 kids.
Yo, today.
Right, yeah,
it's-
look around. No, but honestly, you guys are AP kids, so you may not see it as much. But the amount of kids that just walk into class 10, 15 minutes late every day, there's kids I haven't seen all year, right? And they just don't come anymore, they're just apathetic, and we don't have any recourse. There's no consequence for those kids not showing up, right? They sure, they fail, but they don't have their parents don't get busted anymore. We can't, if they do show up, we don't have the power to give them a detention, right? And if we give them a detention, we can't force them to serve. So there's no reason for a kid to change their behavior. Well, and then we don't get paid when kids don't show up. That's how we get our money from the state, is based on attendance. So when they don't show up, the school doesn't get money. If we don't get money, we can't afford teachers. It's their rotten
So
attitude.
basically, it's our fault.
It's not all the kids' fault, it's not all the attendants' fault, right? We've gotta have some reason for the kids to want to be here in the classroom. If we don't offer,
But at the same time,
if we don't offer,
I'm not here, teaching is what it is.
like marine biology or something that kids wanna take forensic science or something where it would be, some interesting stuff. Do something to get them into the building, dude.
They don't want
And
a high
keep them
school
there.
degree?
What's that?
They don't want a high school degree?
Some people definitely.
Some people think, you know why?
Some
Because,
people just
because
grow
they
up
watch,
like
they
that.
watch TikTok and they see these influencers, I never went to college. Great, you're 1 in 200 million that isn't on government subsidy without your high school diploma, great. Way to set an example.
Are they going to the court, not
The hearings?
Yeah.
No,
Oh.
Why?
because my job's not in jeopardy.
Wait, so
Yeah,
you're
we switched
teaching next
teachers
year?
for that.
Here?
I've been, yeah, I can be here again next year.
Are you gonna teach in bio?
Fine.
My friend wants to teach bio.
Oh, okay.
I'm trying to see how many.
I think there's only,
It's like-
there's only 20-something kids signed up for AP Bio next year.
That's good.
Yeah.
I mean, that's one of the
That
things
means
they were gonna sign up
this
for.
will go quicker and stuff, we can get more labs in, stuff like that.
Lucky.
Okay, let's
Lucky.
not make it
Alright.
lucky.
Bro, what did I
I
do
need to be in
during
this
this time?
class.
I should've waited.
I need to be in this
Dispersive model. Material in the two parental strands is dispersed randomly between the two daughter molecules. After one round of replication, the daughter molecules contain a random mix of parental and daughter DNA.
It's also kind of the school's fault, because all like the older teachers that nobody likes, and we had good teachers, they like took away the good teachers.
So, and,
Like Courtney, like,
right,
Courtney taught us so well.
yeah.
This thing, like, all of them are things.
So, and that's nothing the school has any power over. Because teachers are a union, there's rules that the district has to follow that our union sets up in the contract. And one of those rules is seniority. I can be the worst teacher on campus, or in the district, I could be the worst teacher in the district, but if I've been here the longest, you can't do anything to me.
Wow.
You have to get rid of that younger teacher that comes in here passionate, wanting to coach sports and host clubs and attend dances. Well, you gotta let that one go and keep the old, crusty,
Sack of meat.
yep,
Oh,
you have to,
damn.
you have to. And it's really hard to get them dismissed.
I was gonna take physics before doing.
Yeah,
Yeah, they don't even have AP physics or honors physics next year. They only
Yeah.
have regular physics.
I think they'll be
No, but-
blended again.
Yeah, because like even all the regular places, kids are struggling because of the AP things.
So, which model is correct? Here's what we did, is we took, nitrogen, 15, which is an isotope of nitrogen, right? So we can follow it, because it has certain characteristics, and we got to see how much nitrogen 15 was in daughter cells, right? So bacteria, heavy isotope, nitrogen 15, transferred to a medium with nitrogen 14, a light isotope. This way, if the new bacteria are blended, nitrogen 14, 15, right? Even distribution or whatnot, or if it's all nitrogen 15 in one cell, all nitrogen 14 in another cell, then that would be conservative. Right? By looking at them, it would tell us what was going on. DNA centrifuged and analyzed after each replication. Centrifuged means that we put it in a little tube and put it in a machine that spins really hard, and it'll separate things by density. So nitrogen 15 is more dense than nitrogen 14, so it will be here, and we'll see color bands, and that's how we know what is where. What is for tomorrow?
Yeah.
I wonder if do you know who you have?
No. You know who she has.
I mean, but it's my goal every day to make one of the kids cry.
Oh, why?
Why? Because it's funny.
I hope I don't have you.
Oh, I'm gonna request you, Scarlett.
Yo.
Yeah.
That's crazy.
If you don't complain, you can say you were trying
I
to.
like the funniest part. The funniest part is when they're sitting there and you're going through this, and you just rip it out like trash. Trash.
You don't do that. You don't do that.
Yes, I do. Ask
No, you don't.
Mr. Benjamin. I had
No
him
way.
last week. Last week, him and I were interviewing people together.
Mr. Bunch?
I told Mr.
How did you get stuck with such a nice teacher?
Oh my god!
That
They said that
is
they
so
were good
evil.
Is that?
Thank god I didn't have you.
Oh, did you go last week?
Yeah, I
Who'd
have
you-
my coach.
Who'd you have?
I have Medina.
Medina?
Yeah. So he was in there defending me.
Yeah, good.
Do you ever fail kids when they do their class on
I never fail kids. Kids fail themselves.
Oh.
I'm always
That's
there
not-
for them.
That's a lie. That's a lie. Who told me
Does it
that?
happen
There's some
often?
things in here that I think I should-
The only time I have scored harshly on a capstone was, it was actually the first year I was here, and two twin brothers came in, one right after the other, and they just copied.
Oh my god.
So they even had the same, they didn't change names, like I'm so-and-so. No, you got your mother's name in there, dude. Yeah, that was brilliant, dude. That's the only time I have had a harsh grade on a capstone.
You just said it was brilliant and you graded harshly.
Yeah. I was being sarcastic. Semi-conservative. Analyzing samples of DNA after each generation was found that the parental strands were following the semi-conservative model. So the densities dispersed, and found out that, hey, you get one old, one new.
You can't do anything in the classes, there's like 5 people. It's so quiet.
Like I could fart and like everyone would hear it. Like actually.
It's not funny.
We good?
Wait, no.
Oh yeah, attack is great.
Hurry. That'd be funny to go through this whole entire lecture today. That would be great.
You should have like a test for this.
Trauma.
Right, give you guys a quiz tomorrow on today's lecture.
Yeah.
Yeah.
But like not have us do.
That would be so dumb.
I'm just I know we're not doing it. So.
Are we good?
Yeah.
Semi-conservative model. So generation 0, we had this much nitrogen 15, right? That's what the bacteria had. And then we put it in a medium with nitrogen 14. Why put it in a medium with nitrogen 14? Because that nitrogen would be taken up in making new DNA. We need nitrogen, right? Or nitrogen bases. Our nucleotides. So in generation 1, then it was like this. Cool, we got one old, one new, so it was evenly distributed. Now look, Yeah, you're not getting this old broken up anywhere. You're still getting one old, one new, one old, one new. These guys are still 14. Why are they still 14? Because that's what they're being grown in. That's the only nitrogen that can be used to make it. If it were being nitrogen 15 at all, it would be splattered around in here. So even after 4 generations, we still have the same amount of nitrogen 15, and then all of this is 14. That's what we saw. That tells us it's semi-conservative. The 15 never got broken down. Steps in DNA replication. We have a 3' and 5' end, right? This is what we've talked about before. We read in a certain direction, and we start at what we call the origin of replication. Multiple origins of replication can be opened up on a single strand of DNA at a time. You can have this origin of replication open up, you can have one over here, you can have one over here. We don't start on one end of the DNA and have to go from one end all the way to the other. We have multiple pieces being replicated at the same time. It speeds up the process of replication, so we don't have to wait. Right? You can get all these things going off at once. Various proteins attached to the origin of replication and open the DNA to form a replication fork. This is the fork. Right? Just like a fork in the road. It's just a split. Right? So this is the replication fork right here.
It does.
Now that you said that, I'll crack it up.
Wait, can you go back?
I can.
Please.
I would never write replication fork again. I would just write RF and circle it or something.
Yeah, but not everyone understands those terms.
Oh. Watching you write is like listening to Stephen Hawking.
Who's that? Damn.
Wait, who's that? I almost swore.
What do you mean you're old if you did?
You gonna show her Stephen Hawking?
Who's Stephen Hawking?
I actually don't know. I don't know the reference.
You know? You ever heard Stephen Hawking's talk?
No.
She just got to
No.
Who is Stephen Hawking?
Oh, I can.
Yo!
I can show you audio recording.
Yo!
It's bad.
Listen to him.
I know. I'm just not. I'm just kind of.
Well, I know it's a little speaking spell.
Oh.
Watch, do a video of. That's what it's like watching you write units.
Don't worry about it.
You're about to hear it right now.
No.
Oh, bad.
Let me listen.
The system was put together for me by David Mason of Cambridge Adaptive Communications.
What sounds like an
I run a program called Image.
He could just use Siri.
He can't talk.
He can't talk. He has a talk button.
He doesn't even press buttons, dude. It's like thinking and his eyes do it.
That's crazy.
Well, he's dead.
But what is he? What is he talking?
What is he screaming?
He was a brilliant physicist.
What? I mean, who?
Either
Someone
case.
who's disabled.
Huh?
Bad.
You just called me like disabled.
No, I didn't. I didn't say you were.
I just want to write neatly, so I write slow.
So helicase, its job is to unwind the DNA strand at each replication fork and unzip it to help keep the DNA from rebonding. So you got to think about this. When that DNA separates, you've got your A's and your T's that were once together and now they're separated from each other and they're not happy. They really want to be hydrogen bonded to each other. So to keep the DNA from snapping back together, the single strand has single stranding binding proteins. These attach to the open pieces of DNA so that they don't slap back together. Keeps the DNA pried open. Topoisomerase. is what prevents the strain ahead of the replication, or it relaxes the strain or the DNA ahead of the replication fork.
Wait, go back.
Topoisomerase. Single strand binding proteins. Helicase. That's it. That's all you need.
How do you pronounce that?
Topoisomerase.
That was not.
Topoisomerase.
Topoisomerase?
Topoisomerase.
Just look back into it.
Oh, Mr. White.
Come on, dude. I learned that word in second grade.
No, you didn't.
No, you didn't.
No, you didn't.
No, you
Stop
didn't.
lying.
Bro, second grade, you were smoking.
Alright, bro.
I was not.
Oh,
You were inherently like pooping.
Damn.
Second grade, you were inhaling
No
cocaine.
chill.
That wasn't great.
Oh my god. It's just funny coming from
I know, right?
Why would you say it?
Hangs on to the wrong information.
That's crazy.
This is why you get bullied.
Yes, you did.
Ellie, this is why you get.
It's the way you always get bullied, bro. That's fucked up.
Oh, fail.
We
Well, they
good?
can't help themselves.
We good?
Oh,
Yes.
will prevent stream. No, why would I do that?
Oh, yeah, good. Oh, Val, let's keep going.
Yo!
Okay, I got it super quick.
Alright, so DNA helicase comes in, unzips, right? So it's breaking these hydrogen bonds. The single strand binding proteins attach and keep these guys from closing back up because they want to be bonded to each other the way they're supposed to. Topoisomerase comes in ahead and relaxes this so that helicase can break the bonds. Make sense? Yes. Is that it? Did you have to add anything to a diagram or something?
Oh no,
Yeah.
wait.
Yeah.
Okay, we're good.
Let's stop helicase.
Go back.
Helicase and then topoisomerase.
How do I
Okay,
make
it's right minute. Here,
Yes.
Oh, it's this one.
Oh.
Yes. You didn't
Here.
even fill out
Oh.
genius.
I'm
Oh.
dude. There's a reason why I did recording.
All right.
I'm taking.
The enzyme primase
Okay, I got it.
initiates replication by adding short segments of RNA called primers to the parental DNA strand. So you have RNA primase and DNA primase, right? So we need a little, a primer gets things started, right? A primer, like in a motor or something, is something that gets the motor ready to go, right? So think of that here, primase, it primes the DNA to be copied. And it starts with a little piece of RNA, now it's primed, now we can start writing DNA. RNA primase is gonna end up falling off, and so is the RNA primers, but we'll get to that. Enzymes that synthesize DNA can only attach new DNA nucleotides to an existing strand of nucleotides. They can't just start arbitrarily putting them together. It has to be at a pre-existing strand. Primers serve as the foundation for DNA synthesis. Without the primer, we can't conduct DNA synthesis.
This morning I woke up with my right eye swollen. Cause my cat was wandering out at night and I'm allergic to my cat. And then like on video, like him just like walking around like where I was sleeping. So my dad wants cats and he cared like, care for his kids.
That's right.
Yeah. How dads are.
That's how we roll.
Yeah.
It's all about.
Oh.
Are we good?
Okay. No.
Oh.
We're bad. You know when people are like stop? I say go, right? Like when someone's like, stop! And I'm like, go! And then look at me like I don't get it.
We're all
Hey,
passing.
are you still
I'm
talking
not
to
passing
Orgy?
this out.
Yeah, I went out with her yesterday actually.
How's she doing?
Yeah, we went to go celebrate her brother's birthday.
How's her mom doing?
She might be diagnosed again.
Cause she was in remission, right?
She was fine, so like the cancer went away, but she before all of that she had cancer in her nose, and like they think
Like
it's coming
skin
back.
cancer or
No,
inside?
inside her nose.
On the cartilage or?
I'm not sure, but they're thinking that she has it in her nose.
Tell her to ask about it. Hope she's doing okay.
I'm gonna see her mom today too. So.
Alright, so steps. RNA primer comes in and attaches. Right? At the pay attention to the primes. We're at the 3' end. RNA primer comes in here. What happened? It's going to move in a certain direction, right? We'll look this. The anti-parallel elongation. This is, I'll go back to this other slide here in a sec, or, yeah, because I don't think I have it here. DNA polymerase 3 attaches to each primer on the parental strand and moves in the 3 to 5 direction. If you look right here, it's on this and it's going to move in the 3 to 5 direction. Remember, these guys are anti-parallel. On the other side of this over here is a 5'. On the other side of this over here is a 3'. So that this is going to go in the 3 to 5 direction. This is going to go in the 3 to 5 direction. Anti-parallel, writing in 2 different directions. Right? This one, the top, because that's the 3. Don't always be like, oh, the top one is the, no. It's gonna go in the 3 to 5 direction, so it has to be read this way. It's heading towards the replication fork. This is going to be the leading strand. This, heading away from the replication fork, will be the lagging strand.
The purple thing?
What?
So there's gonna be
We'll get to it. It'll make more sense. It's on other slides. I'm just prepping you for when you hear that information. Alright, so moves in the 3 to 5 direction. As it moves, it adds nucleotides to the new strand in the 5 to 3 direction. Holy shnikers, Mr. White, you just said 3 to 5, now you're going back to 5 to 3? Well, if this is anti-parallel, and this is 3' here, what does this have to be on the primer? On the RNA primer? It's anti-parallel, this has to be then,
3 to 5.
this is 3' up here,
Oh,
this
5.
has to be 5'.
Yeah.
So we read 3 to 5, we build 5 to 3.
Oh.
So this RNA will be 5' and it's gonna end up 3'. Why is it gonna end up 3' over here? Because the other strand of DNA is gonna be 5' up here. Why is it 5' up there? Because it's 3' over here. Yes, no?
Why is the red thing 5'?
Because it has to attach to 3' and they have to be complementary to each other. So if this is 3', the purple is 3', the DNA, this has to be 5'.
Like if it was on the other side and then it was, it's
When
5',
it moves
then it would
outside,
be 3'.
becomes 3'.
Yeah.
Yeah.
Right.
Alright, so it's not gonna move to the other side down here. Another one is going to come in right here
Well,
and
like,
read in this
but
direction.
if it was-
So that this is 3', this bottom one down here on the other end is 3',
Yeah.
and it's gonna go the 3' to 5' direction. Because it's 3' here, it's gotta be 5' here, so that it can be 3' here across from the 5'. We'll clear it up, dude.
Okay.
But, so we, just keep in mind, you go from 3 to 5. As we're reading, you read 3 to 5, but you build 5 to 3.
Oh, okay.
So read 3 to 5, build 5 to 3. It'll clear up, dude, I promise. The DNA polymerase 3, that follows helicase is known as the leading strand, and it only requires 1 primer. We're gonna have to because we're building the lagging strand we have to, in a different direction, we have to build it in chunks or segments. Which we call Okazaki fragments. I just made that up. I don't even know if that's the word, Okazaki.
Okazaki.
Okay, it is.
What?
What? The DNA polymerase III on the other parental strand that moves away from the helicase or away from the replication fork is known as the lagging strand and requires many primers. So it has to build in chunks, and each time, remember, it can't just attach DNA, it has to have something already there. So that's where that RNA primer comes in, right? So DNA polymerase III, we'll put that, we get that primer in there, and we can build the DNA, attach it to the primer. So this next picture should help clear that up. It would help if I looked at the notes. Oh yeah, there we are. Okay?
Yeah.
Ready to move on? Good. Alright, so we're going in the 3 to 5 direction. Right? If you think about it, this DNA, again, so if this DNA were just in a ladder form, it was all, right, not all twisted up, and we have 3', you understand that 5' is here because they're opposite of each other, right? Well, and that means this over here is 5', and this is 3'. Make sense? We read this direction, 3 to 5. So this has to be read this direction, 3 to 5. The rule is still the same. Because this is 3', what attaches here has to be 5'. Right on? And so that's why we build 5 to 3. We're gonna build 5 to 3. As this guy moves down, right, and we're gonna read, tick, 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 start adding nucleotides. Because we have to read the 3 to 5, well, this has to go like here, and then another one here, and that's 3', so this will be 5' here, and this will be 5' here. That's what we're talking about there. When this meets up with this, a little piece has to be taken off, and we like weld those pieces of DNA together then, right? And we'll get to that. But that's what we're talking about with this 3', 5', reading, writing, all that stuff. So the leading strand, again, going towards the replication fork or towards the DNA helicase. The DNA polymerase III moves 3 to 5 following the helicase. So from the 3 to the 5. We're gonna keep going. The lagging strand, again, 3 to 5. It's gonna go away from this DNA helicase, away from this leading strand. Again, it still moves in the 3 to 5 direction.
Wait, go back. The bottom way?
Go back.
Yeah.
I'm not sure I'm understanding how, like, this even gets here. Does that make sense?
Yes.
Because isn't the rest of this still, like, it hasn't been amended yet, right? So,
Sure. Right. This is here, and then DNA polymerase comes in here, and it just attaches this little piece of RNA. That's all we're doing so far.
oh,
That's it. We're not even actually making anything yet. It should make sense here in a sec. the leading strand synthesized in one continuous segment, but since the lagging strand moves away from the replication fork, it is synthesized in chunks. This is what I said earlier. Those chunks are called Okazaki fragments, segments of the lagging strand.
Oh, you made it up on this, right?
I did. I'm a good guesser. Okazaki. Look at the words you learned today. Topoisomerase, Okazaki, helicase. It's fun. I just throw these words out in random conversations, confusing people that this is like at the grocery store, talk to somebody and then just bring out Okazaki fragments.
How do you even speak that out? Like, how do you mention that?
They don't know what I'm talking about, I can mention it in anything.
Why? What is your purpose?
What is that purpose?
What is your motive?
Because I need to continue to make people feel inferior to me.
Oh my god.
It's a guy.
Yeah.
Get a little
Okay.
lazy.
Smartest guy in the room.
Alright, what has to make fun of it?
No, I know I'm always the smartest guy in the room.
Alright, baby.
You may be the smallest guy in the room, people.
Right?
That's great.
So again, here we go, so leading strand, each of these little segments are gonna end up Okazaki fragments. Oh, did you see the animation?
Yeah?
What?
What end?
So, this is being read that way, we're gonna, right? So here's our 5 prime. And writing 5 prime to 3 prime, because we're reading 3 to 5. And then, these get built in little chunks. This is an Okazaki fragment.
The red thing or the circle?
The chunk in here is the Okazaki fragment.
Oh.
After DNA polymerase 3 forms an Okazaki fragment, DNA polymerase 1 replaces RNA nucleotides with DNA nucleotides. So we have to cut out the DNA or the RNA and replace it with the DNA. Don't ask me about DNA polymerase 2,
What's DNA polymerase 2?
what I just said.
I had a feeling.
We only mentioned DNA polymerase 1 and DNA polymerase 3.
Is there a reason?
Probably. I don't know what that is. DNA ligase. DNA ligase is another enzyme. It joins the Okazaki fragments, forming continuous DNA strand. So this is where we weld that DNA together that I talked about. DNA ligase connects Okazaki fragments together for a continuous strand of DNA. I'm good. So here we go. These RNA pieces are going to have to be broken off so that the strand that comes in here and the strand that's over here can all be welded together by ligand or ligase. So DNA polymerase 1 is going to remove this RNA and put little DNAs in there. Ding! It's going to happen down here. Remove that RNA, replace it with DNAs, and then weld it together with ligase. This is how we get the continuous strand. So those replication forks, because there's multiple of them, we call replication bubbles. And those bubbles will eventually spread and they combine with each other, and then you have the two new strands of DNA. Putting it all together. Woo! Movie time.
I chose Wix for my business because of its massive scope for
Nobody cares, lady.
functionality and
When they figured out the structure of DNA, 3 theories came about as to how it actually makes copies of itself. The first is semi-conservative, conservative, and dispersive. Watson and Crick actually believed in this. They believed that DNA would split in half, and then you'd copy new strands on either side. But there were other scientists who believed in a conservative theory that first DNA remains intact, and it kind of makes a photocopy of itself. And then some believed that there was kind of a combination of conservative and semi-conservative, that chunks of it were being split between the two. And this had to do with, they thought, the histone proteins and how the DNA wrapped around it. And so basically the whole thing was figured out through the Michelson-Stahl experiment. Basically what they used was 2 different types of nitrogen. Good old run-of-the-mill nitrogen-14, and then nitrogen-15, an isotope that's heavier than nitrogen-14. So basically they bred a bunch of E. coli on nitrogen-15 until all of their DNA was nitrogen-15. They then put them on a broth of nitrogen-14, and basically in that first generation, if it would have been conservative, we would have had one band that would have been totally heavy at this N15 line, and then one that was at N14, but instead we got this intermediary amount of DNA. In other words, it was a mix of the two. And then through generation after generation after generation, they were able to figure out that this is how DNA copies itself. It copies itself semi-conservatively. And so to look at that in a little better detail, basically here is your DNA, it's a double helix. It will unzip in the middle, so you can see it's unwinding right here. And then we're gonna add new strands on either side. So we eventually start with, we start with one strand, and we're gonna end up with 2 strands. Each of these strands are identical to that first strand, and each of them are going to contain half of that original DNA. So again, it's semi-conservative in nature. Before we actually talk about the process of DNA replication, we should talk about DNA and the parts of DNA. Remember, DNA is going to have 3 parts to it. We're basically going to have a sugar. That would be this deoxyribose sugar right here. I could circle it. This is going to be our deoxyribose sugar. You're going to have a nitrogenous base attached to that. And then you're going to have a phosphate group. And so there are 3 parts to every nucleotide. Again, we've got a sugar and a phosphate and then a nitrogenous base. But you can see that there's going to be another nucleotide right here and another nucleotide going to be found right here. So let me clean that up a little bit. Because it's anti-parallel in nature. In other words, DNA runs next to each other, so it's parallel, but it's anti-parallel in nature. In other words, the 2 strands of DNA are actually running in the opposite direction. And when I mean running, I mean chemically running in either direction. So basically, how do we tell which way it's going? Well, we do that based on the sugar. And so if we look at this sugar right here, the sugar is going to have a carbon here. So we call that carbon the 1' carbon. It's going to have a carbon right here, and we call that the 2' carbon. It's going to have a carbon right there. We call that the 3' carbon. It's going to have a carbon right there, called the 4', and then it's going to have a carbon right here, and that's called the 5'. And so basically, if you look here, that whole thing is going to run from the, let's look way down here, 1, 2, 3' end. So that's going to run from the 1, 2, 3' end right here, all the way up to the 5' end over here, because here's that 5'. If we look on the other side of the DNA, you can see it's running the opposite direction, from the 3' to the 5'. And that's going to be important when we look at DNA and how it copies itself. And so let's look at that. If we look on the next slide, in DNA replication, there are tons and tons and tons of enzymes that are helping out. It's way more complex than this, but from a diagram level, this is pretty good. So basically, the DNA is going to be a double helix in this direction, but we're going to have this enzyme right here. It's called helicase, and it's basically going to unwind the DNA. So we're going to go from this double helix to these single strands of DNA on either side. These strands are going to be held in place using these enzymes. They're called single-strand binding proteins. They're basically going to hold it in place. And so now we have that unwound DNA. The big enzyme that's super important in here is called DNA polymerase. So if we look on this side, DNA polymerase is going to race down the DNA and it's going to add new nucleotides on the other side of the DNA. So here's the original strand. And you can see that DNA polymerase has already been here because it's added new strands in this direction. Now the trick is that we can only add new nucleotides on the 3' end. We can't add it on the 5' end. And so basically, again, so here's the 5' end. If we follow that right down here, we can add DNA on this side, on the 3' end, and it's just going to go on silky smooth. In other words, helicase unwinds it, DNA polymerase adds the new letters. And on this side, we call that the leading strand. Everything's going to be perfect. It's just going to flow on there perfectly. But the problem is, since we can only add DNA on the 3' end, we can't add it up here. We can't add it on the 5' end over here. And so what's evolved is this really elegant method called the lagging strand. So we can finish out the other side. And it's lagging strand because it tends to lag behind the other side. If you've done any sewing, which I never have, it's kind of like back stitching. In other words, you're going in this direction, but you're back stitching the way as you go. And so basically there are a number of different parts that are found in here. First thing that we have to do is we have to put down a primer. And so there's going to be RNA primase, and primase is going to add down a primer. A primer is just one little bit of RNA. So we'll add a little bit of an RNA first, and after we've added that RNA primer, then DNA polymerase can go in this direction. So once the primer's in place, then we can run in that direction, we can run in that direction, and we can keep running in that direction. So we've got to put a little RNA down, then DNA polymerase goes. Unfortunately, it can't connect it here because we've got DNA bumping into RNA, and so there's going to be another enzyme. And that enzyme is called, let me find it, DNA ligase. And so basically what DNA ligase is going to do is it's going to go after that, and it's going to clean up all these messy junctions here, and it's going to put DNA straight across it. And so basically that's a lot of stuff going on. What is all of that doing? It's making sure that that message that was found in the DNA is copied to that 2 new strands of DNA on either side. But we got to finish. And so basically what I want to talk about is origins of replication, or where DNA replication starts. Well, in life, there are basically 2 life types. We've got the prokaryotics, which is going to be the bacteria and the archaea, and then the eukaryotics, and that's going to be like you. And if you're prokaryotic, you're going to have a single loop of DNA. This is actually a plasmid, but it looks the same way. You have a strand of DNA in a perfect loop. And so for them, they can just simply start copying it on this side. The origin of replication is at one point. They move around, and eventually what they'll have is 2 strands of DNA. It's going to be an exact copy of that. And again, in binary fission, those become different cells. But in us, we have such a long DNA that we have to wad it up to even get it to fit in a chromosome like these pictured right here. In other words, your DNA in a cell is going to be like that long. And so if we were to start on one side and start copying it, it would take forever. And so basically what happens is we work in 2 directions. So basically there will be a site of replication where it starts here, but we're going to have it moving in this direction and moving in that direction. And so basically that diagram that I just showed you, I think this would be a better picture.